Hello, I'm Seth Cooper from the Corey College of Computer Sciences at Northeastern University, and I'm going to talk a bit about Foldit mostly and the origins of scientific discovery games in molecular biology. So a lot of my work looks at trying to use video games for crowdsourcing citizen science and scientific discovery, and basically trying to build games whose mechanics and rules are somehow based on a real world problem, and use those games to try and put the time that people spend playing towards video games, put that time towards solving real world problems, in particular problems that are hard for computers to solve, problems that are computationally challenging, and try to involve human brain power towards solving those problems. And it's mainly for problem solving. It can also sort of be a means for people to get involved in areas of science that may be of interest to them. So, right, like a citizen science kind of approach. So really trying to, combine computational power and human brain power together to solve challenging real world problems through video games. And so one of the kind of high level questions is, how could we use a video game to solve a challenging real world problem? And I kind of see it as trying to build a system that both engages the players and trains them to improve, to work together with computers and improve sort of this whole system over time, make the games become a better problem solving tool, and sort of as the player community can become better problem solvers. And games I think are good at doing this kind of thing, like being engaging and also teaching players how to do new kinds of things in the game that, we can teach them how to do things that might help solve a real world problem. And, try to build something like this, a game like this, and have it get better over time as the players play the game. So the main example that I'm going to talk about here is Foldit, which is a game around proteins, protein folding, and design. I've been working on it for several years now, along with a lot of other people on the Foldit team. And so this will be the main example that I'm talking about today. I'll start out with a project overview, a little bit information about the game in general, talk about some of the player accomplishments that have been possible through the game and with the players, talk a little bit about and a little example of data-driven game design that we do, and trying to use data from gameplay to inform the design decisions that we make, and talk a little bit about some of the ongoing and future projects related to Foldit. So, right, Foldit is all about proteins. They're important for many functions in life and how things get done at the cellular level often. But one of the important things about proteins is their structure determines their function, and so it's very useful to know the structure of the protein in order to be able to understand their structure. But of course, the gap between kind of known protein structures and known protein sequences, right, is growing all the time, and so it's it would be very helpful to know the structure for many of these protein sequences that we have. And there have been a lot of sort of purely computational approaches to trying to compute protein structures, right? Some examples being Rosetta at Home, which is actually both of these, Rosetta at Home and Folding at Home, are distributed computing projects. They're screensavers that can run on your computer when you're not using it, and donate that spare computing time to trying to crunch the numbers on protein structures. Folding at Home, I believe at one point, was the most powerful distributed computing cluster in the world. And even with all of that, spare computing power, the problem is still quite challenging, right? But one of the things that we noticed was, right, and these are visual, right? You can actually see how the protein moves around and how the computer is searching and that kind of thing, right? And it's difficult computationally because there are just so many possible shapes that a protein could fold up into. But people who are running the screensaver could actually see how it's folding up, and people who are running Rosetta at Home could see how the computer is trying to figure out how the protein folds, and see the kinds of moves that it's making and maybe think that, they could look at the structure and think about or figure out how the pieces could fit together a little bit better. And so it's really, right, in a way, one can consider kind of these protein structural problems to be kind of spatial reasoning problems or kind of puzzles, right? It's about the shapes and how they fit together. And people have a really great ability to kind of reason about shapes, how they fit together, how puzzles fit together, and how pieces kind of fit in 3 dimensions. And so we were kind of interested in what would happen if we could allow humans to manipulate the protein shapes or get involved, interact with the computational search, right? So if humans could apply their spatial reasoning and intuition in addition to the purely computational search, maybe they could do better than just computers alone. And in fact, a little bit before Foldit came out, there was another game called the ESP game with the goal of getting people to help out with solving a real world problem through playing a game. The ESP game was based on image labeling, and the idea was that you would sort of randomly pair up 2 people on the internet. They couldn't talk to each other, but they saw the same picture, and their goal was to type in the same word. And so they would type in things that were in the picture to try to type the same thing as someone else. And if they matched, they would get points and move on. And so there was a little bit of precedent for basically, trying to build a game that would get people interested, engage people in helping to kind of solve this kind of real world problem. And so to work on this, we've been developing Foldit, which is an online citizen science game where the players compete and collaborate to find well-folded and well-designed protein structures. The players do these high-level spatial manipulations of the structure, clicking and dragging, pulling on the structure with bands and things like that, freezing parts. And the computer can take over and do some of the low-level optimizations and refinement of the structure. And so it's really set up to try to let the players do what they're good at, high-level structural rearrangements, and let the computer do kind of the low-level pieces. It's built on top of Rosetta, the molecular modeling software, the same software that the Rosetta at Home screensaver is on. And so that's there to help provide sort of scientific accuracy of the proteins. And we've been working on protein structure prediction and protein design in the game. It's been out for over 10 years, had over 500,000 players, and it started at the University of Washington, but now there are a number of institutions collaborating on it, including the University of Washington, Northeastern, UC Davis, Vanderbilt, UMass Dartmouth, and the University of Denver. So players' goal in Foldit is to basically get a high score, and the score that we get is the Rosetta energy, the energy of the structure that we get from Rosetta. And so we just take that energy and we sort of invert it and scale it up. So instead of trying to get a low energy, the players are trying to get a high score. And since we don't really know what the best score is, we organize the whole thing as a leaderboard competition where players are competing against each other to get the highest score possible. So, unless you're in first place, there's always a higher score to try to achieve. And in addition to individual players, there are actually teams that can compete against each other. So within the teams, the players are working together to get the best score they can get as a team. And so there's some collaboration going on, but then the players themselves are competing and the teams are competing. So there's competition and collaboration going on. We really wanted to try to make the game approachable to players who might not have a biochemistry background. So we have a series of introductory tutorial levels that are separate from the scientific puzzles that are meant to teach how to play the game to new players. And they start off with relatively simple structures with relatively simple things wrong with them, teach how to use the tools in the game to improve them, and they have set goal scores so that players can complete them and move on to the next tutorial puzzle. And as players progress, the tutorials, get more complicated to the point where hopefully a player who completes the tutorials would be able to compete in the scientific puzzles. And we've seen that a lot of the players who are doing well in the game actually don't have any formal biochemistry background. So next I'm going to talk a little bit about a few things that players have accomplished in the time that the game has been out. Foldit players are actually listed now as authors on 2 structures in the PDB. shown here, and I'm going to talk a little bit about the first one. I believe for us, a little bit later today, we'll be talking more about the approach used in the second one. So one of those structures came from some of the early work that we did in Foldit in protein structure prediction. This was related to the Mason-Pfizer monkey virus retroviral protease, which is plays a key role in simian AIDS in rhesus monkeys, and experimentalists who we collaborated with had worked on it for over 10 years, they told us, and sort of the recent computational methods had failed to solve the structure. We gave it to the players for 3 weeks, so we gave them kind of this red starting structure to start from. And it was actually one of the interesting things. They were able to solve it. The players were able to solve it in less than 3 weeks. And in fact, one of the interesting things was it wasn't just one player who was able to solve it by themselves, but it was actually 3 players working together who were able to come up with a structure sort of good enough quality that it could be used for molecular replacement. And so these 3 players sort of collaborating in their team were able to come up with a very good structure. The blue structure here, actually, now that we know what it is, the native structure, you can see how the players were able to take this starting structure that we gave them and sort of by working together and improving on each other's work, get closer and closer to the structure that we were able to use to solve this. Recently, we've been working a lot on protein design, which I think is really exciting because we can actually take the designs from the players come up with in the game and bring them out into the real world and do some experimental validation on them, right? So the players can sort of computationally design protein structures in the game and see how well they do and compete and collaborate with other players to see who can do the best within the game. And then we can actually, test some of those designs in the lab, in the real world, and see how they work. So a recent example that we've been working on in this area is de novo protein design with the players. So giving the players basically an extended chain of a protein and letting them design whatever they want to see what they can come up with. In fact, then we can take some of the designs that the players come up with, filter through those to try to find some promising designs that come out of the game and select a few of those structures for testing, and then synthesize, crystallize, solve the structures in the lab, and see if they can be solved and we can get an experimental structure out of these. And so this is actually a crystal of a player designed protein here, and see if the experimental structure actually matches the structure that the player designed in the game. And at this point, we actually have had 4 out of this project, right? 4 player designs that came out of these sort of de novo protein design puzzles that we ran in Foldit. And a lot of this work was based on trying to help players design realistic structures by updating the in-game scoring function to better award protein designs that have good protein features, kind of good basic protein features such as nice cores and things like that. And in fact, based on some of these player designs that were made in the game, we wrote this paper. A lot of this project was led by Brian Kepnick, and the players who made these designs in the games, they actually ended up as authors on a Nature paper, which I think is actually really a really exciting kind of thing. Also to come out of this is not just the structures themselves, but involving citizen scientists in the process, in the scientific process, and them getting to be authors on the paper that was written about the structures that they designed. And so next I'll talk a little bit about the data-driven game design approach that we use in Foldit and how we were able to, over time come up with this game that allowed players to contribute to the scientific process in this way and make the game a sort of a better and better problem-solving tool, and help to make the game a better way to sort of like teach players about protein folding, hopefully. So it's really an iterative process that we're running, where we have scientific problems that we're interested in running, be these structure prediction problems or design problems, and we work with scientists on the team or collaborators to basically take the problems that they're interested in and turn those into something that the game can understand, which we call puzzles. And puzzles, they have things like a starting structure, whether you can design things or not, what the energy function we're using is, and so on, and things like that. And then those puzzles get distributed out to all the players who are playing the game. And in fact, these usually run for about a week. Each puzzle is usually up for about a week, and players can access the puzzle during that time and compete, collaborate, try to come up with the best structures that they can based on the parameters of the puzzle. And then when the puzzles close, we can analyze the data from the gameplay, looking at the kinds of structures that the players found and how they found them, and see if there's anything interesting or useful that they found in there, or maybe we need to make some changes to how we present the puzzle or the game itself. And so it's really been an iterative process over the course of the project to try to continually improve the game and the kind of the quality of the structures that come out of gameplay. And in fact, in addition to the sort of the protein structural information we can get information about how players are playing the game, in particular in the tutorials, to try to make the tutorials better and help new players come in and enjoy the game and learn the game better. So that's something that we're also always trying to improve. One example of this was looking at how we provide help to players in the tutorial levels to teach them how to play and help them learn the game. And so we ran an experiment that compared a couple different ways of providing this help. One of those ways being not providing any help at all. Another being providing what we called context-insensitive help, which was just giving them an instruction manual at the beginning of each level that told them what to do in that level. And the third being what we called context-sensitive help, which had a system that tried to figure out what the players were doing, where they were in the puzzle, and what they needed to do next, and basically tell them what they needed to know right at that point. And when we looked at the number of levels completed, perhaps unsurprisingly, players completed the fewest levels with no help, but they didn't actually get that much further with the context-insensitive help, I think, because oftentimes people will just dismiss this kind of instruction manual help and not read it very closely. But the context-sensitive help provided, we think, the most help and helped players get the farthest through the game. And in fact, when we look at how long players were playing, we see a similar pattern. And so the context-sensitive help is actually what's in the game right now. So there are a few ongoing and future projects I wanted to mention. We're working with collaborators at Vanderbilt to try to put in tools for small molecule design in the game. We're also collaborating with the team at UC Davis to put a sort of a narrative story into the tutorials and see if that makes them more interesting. And one thing that we're often talking about working on is if we could sort of port Foldit into virtual reality and how that would work and if that would actually help players. I'd like to end with just a few ways that Foldit might be able to help you if you're listening to this. We're always looking for collaborators in Foldit, people who might be interested in posting their structures or the problems that they're interested in, and collaborating with Foldit and the Foldit player community to see what kinds of solutions and things they come up with, and there are a variety of ways to do this. So people are collaborating and using Foldit for research and education as well. We also have Foldit Standalone, which is a separate application, but it provides the Foldit interface separate from the game and allows users to import and export their own protein structures to work on. And I'd like to thank the Foldit development team, as well as our biochemistry collaborators and supporters, and of course all the players without whom this wouldn't be possible. You can try the game online or contact me if you have any questions. Thanks.